In this video, we're going to describe exploratory data analysis techniques that are particularly useful for high throughput data. We're going to show some of these on the data set that has gene expression measurements for several genes, several thousand genes. We're going to perform a t-test on each one of these to show our first exploratory data analysis technique, which is the histogram of p-values. We're also going to generate, with the Monte Carlo simulation, a case where we know that for all genes, the null hypothesis is true. So the first technique we talk about here is a very simple one. We just basically make a histogram of the p-values. We'll make this pane a little bigger so you can see these figures better. And let's start by making a histogram of the p-values for the null case. And you can see, as expected, they look uniformly distributed. So this looks like a case where there's perhaps no interesting differences, but also a case where there doesn't appear to be any artifacts causing this distribution to look different than the expected uniform. For the other case where we actually may have some differentially expressed genes, we see that it looks uniform for the first part, but then we have a pretty big peak here showing that there's a lot of genes that appear to be differentially expressed. It turns out that in this data set there is in fact a problem with the data that we are not going to talk about here, but this is one way we would start getting suspicious about it when we see in a case like this where we don't expect that many genes to be differentially expressed. That's knowledge that we get from many times talking with the data generators or the experts, and we see these many evidence for these many differentially expressed genes, then we get a little suspicious. So in this case, the histogram of p-values is actually quite insightful. A second plot that we can make is called the volcano plot. So one of the problems with the p-values, as we have described in a previous course, is that it doesn't give you any idea of the estimated effect size. It just gives you a probability of seeing something under the null. The volcano plot lets us see, it shows us the effect size that we use in the t-test along with the p-value. So it's very simple. You make a scatter plot of the negative log 10 p-values against the estimated effect sizes. The reason we take negative log 10, let's just make that plot, is because we want the high genes to be at the top and the low, the genes that have high significance to be high on the plot, and the ones that have low significance low on the plot. So here we have the effect size, and then we have some genes that are clearly differentially expressed out there and have very large effect size and very small p-values. So that is that's the volcano plot. It's a very useful figure that gives us an idea of genes that are both have small p-values and large effect sizes. You can immediately see it in a plot like this. Okay, a next set of plots that we're going to show relate more to data exploration. And now we're going to load another data set that is much bigger. This one has many more columns. Let me show you how big this is. So we're going to put the gene expression here. Here we're using some bioconductor functions to extract gene expression data, which you can just follow along the code if you have not seen this before. But this one has 208 samples. And what we're going to do is we're going to introduce a problem on purpose. So on the 49th gene, we're going to divide by log 2 of e to the 1. And the reason we're doing that is we're acting as if when they generated this data, they incorrectly took the natural log of one of the samples, and when all the rest are in the log 2 transformation scale. So there's one sample that is where the measurements are a mistake. But we don't. Let's suppose we don't know that. How could we find that? Well, one thing we can do is simply make box plots of all these genes, and there they are. Let's change this guy first. Introduce the error, and now when we make a box plot, we immediately see that there's a problem there, right? So all these box plots are roughly the same, and then there's one outlier that is behaving differently. Now, there is one of the problems with making so many box plots is that you kind of run out of space. They're all kind of crammed together, and we can't really see each one individually. So if you have a lot of samples like we do here, we can instead just plot the quantiles. So here in this plot, we're going to just plot the 5th percentile, 25th percentile, 50th percentile, 75th percentile, 95th percentile for each sample. So we're going to do that very quickly. We just compute that and then use the function mapplot to see all them. And again, immediately we see that the 49th sample is different from the rest, in that every single quantile that we're showing is lower. So if you think about it, this is very similar to a box plot, except instead of making the box plots, we're just plotting the quantiles. Okay, another figure that you can make are smooth histograms. So what we do is instead of making a histogram with the bars, we smooth the top of the bars to get a curve. This is also called an empirical density. And we plot all of them together. Instead of making 200 histograms, we plot them all together, and then we can detect perhaps samples that are very different. For example, here's our problematic sample. It comes out right away that it's different from the rest. Okay, the final plot I want to show you is the MA plot. When we want to explore the relationship between two samples, for example, here I'm going to use 1 and 2, samples 1 and 2. We could plot them against each other, like that. But there, if we make that plot, it looks like it's very nice and replicated. It's very high correlation, it appears. But if what we're interested is in the differences between x and y, a much better plot is to plot the difference in the y-axis. And then what we'll do is in the x-axis, we'll put the average, because that's also informative if it's a highly expressed gene, which will be up here, or a low expressed gene is here. So basically what the MA plot does is plots the difference. In this case, because these are in the log scale, this would be the log ratio. It plots it against the average. And it looks like this. Right, so now we can see the variability is not that small. It's not that big, but it's not that small either. Here it appears to be very tight, but there's actually differences of log ratios of 1 in some instances, a -1 in some instances. So that is the MA plot that's very commonly used. We sometimes discover patterns in the data that aren't seen in the scatter plot, but that we can in fact see it in the MA plot. And of course, the MA plot relates to the it also helps you see that the standard deviation between these two measurements is 0.20. Again, that's not that small, saying that a typical gene shows a log fold change of 0.20. And that's something that's hard to detect from the scatter.